A construction file describes lab operations for fabricating a genetic construct. In this diagram, we illustrate the various steps of the cloning experiment. First, we'll do a PCR with two oligonucleotides named primer 1 and primer 2 using plasmid DNA called pProbe GFP LVA as template. This plasmid encodes the GFP gene in green that we wish to move to another plasmid. After PCR, we'll need to digest the product with EcoR1 and Hindi3. These restriction enzymes will cleave the DNA at the E and H sites. That digestion will result in 3 fragments, a large one with the gene we want and 2 very small tails. In the purification step, we'll isolate the fragment containing GFP. Similarly, we need to digest PUC19 with EcoR1 and Hindi3, and then purify the large backbone fragment containing the origin of replication and selectable marker. Finally, we ligate the GFP cassette and the vector backbone together to make our product, PUC-GFP. A construction file is basically a formal way of expressing a cloning experiment. Minimally, it allows you to explain precisely to your computer the experimental plan for documentation. Also, simulating construction files helps catch design errors and thus saves time in the lab. Best of all, software can automate the drafting of construction files for simple experiments. 2 pieces of information are needed for a construction file: the list of input sequences and the reactions. This list of sequences for our experiment would include pProbe GFP LVA, PUC19, primer 1, and primer 2. And there are 6 reactions: 1 PCR, 2 digest, 2 purifies, and 1 ligate. In the GUI, these distinct reactions and inputs are displayed visually as a dictionary specific to this project and a list of reactions that reference this dictionary.